Assalamu alaikum. So we talked about transcription, and we said that transcription is the process of making RNA using one of the strands of DNA as a template, and this is done by RNA polymerase. And we said that when we look at eukaryotic genes and human genes, that the RNA polymerase sits on the promoter and it starts transcription at the transcription start site. And it ends at a termination sequence known as the polyadenylation signal. So this here sort of summarizes what I talked about in the past two lectures. In that you have a core promoter. If you look at the DNA and you look at the gene itself, the expression of any gene, of certain genes, can also be controlled by what is known as proximal promoter element, and also by enhancers or silencers. And we said that these enhancers and silencers can be positioned or located far away from the gene, upstream of a gene, close to the gene, within a gene, downstream of a gene, and you can flip them and they're still functional. And the reason is that there is something called DNA looping. It's a feature, characteristic of DNA, that DNA can loop around, and you can have this interaction between enhancer binding proteins and the promoter binding proteins mediated by the mediator. So anyhow, so the gene is transcribed at the transcription start site, and it ends at the polyadenylation site. Okay, and when the mRNA is first transcribed, it's transcribed with the exons and introns as part of the messenger RNA itself, and this is the primary messenger RNA, primary transcript, pre-messenger RNA, whatever you want to call it. Okay, and the messenger RNA contains exons and introns, and these introns are never part of the protein. And exons can be selected. So you can have what is known as alternative splicing, whereby you can have removal of all introns, and you can have also removal of exons along certain exons along with the introns, so that you can produce different mature messenger RNA molecules that can make different polypeptides that can become proteins with three-dimensional structures and functions, specific three-dimensional structures and functions. And you can have what we call as protein isoforms. Okay, so different proteins with a similar function, like actin binding proteins, and so on. And but they differ. They differ in their strength of action, their kinetics, if we're talking about enzymes, their regulation, their effectiveness in sending signals, for example, interacting with other proteins, and so on. So this can be true for many of the 20,000 protein coding genes. And this is the reason alternative splicing is one reason why, although we have 20,000 protein coding messenger genes, we can produce 80,000 protein coding messenger RNA molecules, transcripts. So that increases diversity. All right. Now, the number of proteins is actually much larger than 80,000 proteins. And I'll talk about some phenomena in this lecture. I'm going to talk about some phenomena related to eukaryotic genes, to human genes, whereby we can have limited number of protein coding genes, but we can ultimately produce many different types of proteins. So let's talk about these phenomena now. Starting with the concept or the mechanism of gene rearrangement. So the gene itself is rearranged. So let's talk about that. Immunoglobulins is a perfect example for that. So immunoglobulins or antibodies are proteins produced by B lymphocytes, and they can interact with antigens, foreign bodies, as a mechanism to eliminate these antigens from the body. Now, if you look at the structure of immunoglobulins, they are composed of 4 polypeptide chains. Two identical chains, are known as the light chains, and 2 are identical heavy chains. And they are linked to each other by those disulfide bonds. Now, if you look at the structure of the light chains, they are composed of a constant domain, constant region, and a variable region. Now, if you look at the structure of the heavy chain, it is composed of a variable region and 3 constant, and a constant region, okay, composed of 3 domains. Now, the variable region is the part of the immunoglobulin that is responsible for binding the antigens. Now, how many antigens do we have in nature? We have trillions. We have so many antigens in nature. So immunoglobulins must also be diverse so much that they would be able to interact with all of these different, possibly with all of these different antigens. Now, it's really impossible to have in our genome trillion genes that can produce the different antibodies. So there must be a better mechanism. Now, in fact, in our body, we can produce about a trillion different B lymphocytes. These are the cells that produce the immunoglobulins. Now, each B cell can produce one single type of immunoglobulins, just one. Okay, so we can have this number of B lymphocytes in our body as well. So how is it possible that that B lymphocytes can produce this huge number of immunoglobulins? Well, this is done by site-specific recombination as well as other mechanisms that some of which I talked about in the biochemistry lecture. So let's look at the molecular biology of the immunoglobulin gene. Well, this actually is taken from the mouse gene, okay, not the human gene, but the same applies for the human gene as well. That's one. Now, 2, I'm just going to talk about the production of the heavy chain, but the same exact concept applies to production of the light chain, same mechanism. Now, next I'm going to talk about the production of T cell receptors, but it's really the same mechanism. But I'll talk about T cells later on in the next slide because of a new technology that I would like to talk about. So if we look at the gene of an immunoglobulin, we would find that the variable region can be coded by actually 3 different types of exons. One type is known as the V exons, and there are 150 different exons representing the V region. 150. Now, another type of exons is known as the diversity exons, and there are 12 of them. And the third type of of an exon that can code for a portion of the variable region is known as the joining exons, and there are 4 of them. Now that's the gene of a naive B cell. A naive B cell is the cell that has not been exposed to an antigen and it has not been activated. It has not been stimulated. And it gets stimulated when the immunoglobulin on the cell surface interacts with the antigen. So, when the B lymphocyte develops, what happens is that one of the V exons, one of the 150 V exons, would recombine, would be rearranged so that it becomes part of one of the D exons, and one of the D exons can recombine, can be rearranged so that it becomes part of one of the J exons. So what happens first is that one of the D exons, diversity exons, would be rearranged. So right here in this example, you have diversity exon 5 rearranged, becoming part very close to J2. So this portion right here, J1 all the way to D6, would be removed. It would be deleted from this particular B cell line. Now, and then one of the 150 V exons would recombine. So here it's like V25 would recombine with D6. Now, so everything between V25 and D6 would be totally removed. It would be deleted. Then what happens is that, so you have this recombination right here, and then what happens is that you have the production. Now each one of these V regions, by the way, has its own promoter. So so you have production of the primary transcript. The primary transcript starts with the V25, so transcription starts right here. And so you have transcription of V25, D6, J2, and everything afterwards. And that also includes the C region, the constant region. And then you have splicing happening, so that everything between J2 and the C exon would be removed. Now this is the mature messenger RNA. Now if we consider that we have 150 V exons and 12 J exons, D exons, I'm sorry, and 4 J exons, we're talking about 7,200 different immunoglobulins that can be produced. Well, similarly, same mechanism applies to the light chains, and 600 different light chains can be produced by the same mechanism. So if we multiply 600 by 720, we would get about 4 million different combinations of heavy chains and light chains. Now what happens as well is that when you have joining of the different segments, that is the V exons with the D exons with the J exons, things are not perfect. So you would have the addition or deletion of a few nucleotides, and that increases diversity even further. In addition, during the DNA replication, during the proliferation of the B cells, somatic hypermutations can also take place randomly, and these are random mutations that can take place during DNA replication, and that would increase the diversity of immunoglobulins even further. So we would end up with about 1 trillion different immunoglobulins produced by the different B lymphocytes. But once we have the DNA rearranged, by the way, it doesn't change anymore. Okay, that is it's fixed. That's it. This B cell has this DNA because all of the portions that of the gene that are not needed would be removed permanently. So this is the mechanism by which we can produce a trillion different B lymphocytes. Now the same thing applies to the T receptor, T cell receptor. Now the T cell receptor has a similar structure as immunoglobulins. There's variable regions. There are two chains, alpha and beta. Each one of them has variable regions, and so on. So and the production of these receptors is basically the same, genetic recombination. Now, I want to just mention T cells and T cell receptor because now we have the production. Well, we have this innovation of the production of what is known as CAR T cells. And CAR T cells are basically genetically engineered T cells, whereby the T cell receptor is engineered at the genetic level so that we use it to fight cancer. So T cells would be removed from a patient, they would be engineered in the laboratory, and then the same cells, after being engineered, they are injected back into the patient. And they are engineered in a way by which the T cell receptor would recognize cancer cells. So when they are injected back into the cancer patient, these T cells would be activated because they would recognize and bind to the cancer cells, they proliferate and they remove the cancer cells. And this is the new trend in cancer therapy, CAR T cells. So remember this because this is the current and future technology. Um, and it's great, it's wonderful, good news. So you can watch this video, by the way, if you want to learn more about the CAR T cells and how they are produced. So that's one. Another mechanism is something known as gene amplification. And basically what happens in gene amplification is that we already know that we have genes located, positioned on chromosomes. And a gene is composed of a region on the double-stranded DNA. It's a small region, well, relatively small region within a chromosome, and it's basically double-stranded DNA, and one of the strands would be used for transcription, like I said before. Now what happens, and this happens naturally, is that cells can have multiple copies of that gene, of this region, on the same chromosome. So instead of having one copy of a gene on a chromosome, we can have like 10 different copies. And all of them have their own promoters, and all of them can be transcribed, can be used for transcription. So the chromosome itself would look something like this. It would be puffy, meaning, inflated, like puffy fish. Now, so this happens naturally, by the way, as a mechanism of fighting drugs, for example, or or cells being under stress for one reason or another. Now, cancer cells can take advantage of what is normal, what is natural, what is physiological, and they can take advantage of this and they can utilize this to escape therapy. So cancer cells, you can use it to develop resistance against methotrexate. We talked about methotrexate as a cancer chemotherapeutic drug in the biochemistry course. Now, what happens is that this methotrexate targets dihydrofolate, the enzyme dihydrofolate reductase. because it resembles folic acid or folate. Now, what happens is that what cancer cells do is that they can amplify the dihydrofolate reductase gene, so they can produce a lot of it, and they can overcome the effects of this drug, methotrexate. Something else, another example that cancer cells can do is breast cancer amplifying the human epidermal growth factor receptor 2, or HER2. So normally there is a limited number, relatively low number of HER2 receptor on the cell surface of epithelial cells, mammary epithelial cells. When breast cancer, well, becomes cancerous, when mammary cells become cancerous, they amplify the HER2 gene so they can produce a lot of the HER2 protein on the cell surface and present it on the cell surface. So they become very aggressive. Okay, so, well, scientists and clinicians have taken advantage of this phenomenon of the HER2 gene amplification. And this can be detected by immunohistochemistry and by FISH. So immunohistochemistry is basically using antibodies on tissues, on tissue sections, like you learned in histology. So basically, we have breast cancer tissue, and they can be stained for HER2. If there's a lot of staining, and a score is given to the immunohistochemical staining of HER2. So if there is a huge production of HER2 in cancer cells, then they can be treated with Herceptin. I'll just talk about Herceptin. Now, if the scoring of immunohistochemistry staining of HER2 is sort of like intermediate, if it is what they call it unequivocal, meaning that it is, it's hard to determine if it is positive or negative, if it is, if it can be, if targeting HER2 would be the best suitable treatment for this type of breast cancer, then FISH is done. FISH is fluorescent in situ hybridization. I talked about it in the human genome lecture. So basically, we use a probe, and the probe is labeled, and it's added to a tissue section, and it's labeled with fluorescence, by the way, and then it's added to cells in a tissue section. And normally there should be low signal, 2 signals, let's say, shown in red right here, because in green is basically centromeres. Okay, so you have 2 chromosomes here, and there's 1 staining on each chromosome. Well, same thing for these cells, for example, and these cells, and these cells, and so on. So that's normal staining. But if you see a lot of staining in red for HER2, this means that this breast cancer is what we call HER2 enriched. That is, there's amplification of the HER2 gene, and these cancer cells are dependent on HER2 in becoming aggressive, in having high level of proliferation. And that's why this type of breast cancer, the HER2 enriched cancers, can be treated with Herceptin or trastuzumab. So whenever you hear the word MAB, it means monoclonal antibodies. Go back to the biochemistry lectures, and we talked about monoclonal antibodies before in the immunoglobulin lecture. Trastuzumab, commercially known as Herceptin, targets the HER2 receptor. Now, what happens is that we give patients these monoclonal antibodies, and these antibodies would target the HER2, blocking its function, driving the cells to die, to become ineffective, to prevent the actions of the HER2 and its growth factor ligand. So this is the treatment, and it is an effective treatment for breast cancer. So it is connected to gene amplification. Well, let's talk about a third phenomenon. Here we have a gene that can have multiple exon 1s, and each one of them would have multiple promoters. The concept of a gene having multiple promoters, by the way, is somewhat common in the human genome. But let's talk about this one single example. So we have this enzyme right here in our liver cells, the UDP glucuronosyl transferase. Now, this enzyme right here transfers the UDP glucuronate onto drugs. Now, note that this molecule right here is highly hydrophilic. So what happens in the liver is that some of the drugs are modified enzymatically by several enzymes, different types of enzymes, such as the glucuronosyl transferase enzyme. And what these enzymes do is that they take the glucuronate or glucuronic acid and they add it to xenobiotics, to drugs, making them more hydrophilic so that they become water soluble, and it would be easier to eliminate, to excrete, to remove these drugs from our bodies. Now, what are the substrates that this enzyme can act on? Well, different types of substrates, different types of drugs, different types of chemicals. It can target, the enzyme can target therapeutic drugs, different carcinogens, steroids, fatty acids, bilirubin, different types of molecules, estrogens, for example, and so on. Now, if you look at these molecules, they differ a lot from each other. So the question is, how can a single enzyme bind to all of these different drugs and modify them by adding glucuronate to these molecules? Again, not only that this enzyme can act on different drugs, it can act on these drugs on different tissues. So it is substrate specific and it is also tissue specific as well. So how can such an enzyme do such a thing? Do not memorize this, of course. Well, the concept follows this. So we have one drill, right? And we know what a drill does, like the enzyme. What the enzyme does is that it adds glucuronic acid, right? Now, that's what our drill does. It makes a hole in the wall. But we can connect different flutes on a drill. So it can do a lot of things. Or we can have the same head, but we can wear different hats, changing the function of that person by changing the hats. And the same thing applies to this enzyme. How so? Well, if you look at the gene it consists of 5 exons. Exon 1, and then you have exons 2 to 5. Exon 1 is responsible for the substrate binding domain. Now, exons 2 to 5 code for the catalytic domain. So these exons would be transcribed into eventually a mature messenger RNA, and these exons 2 to 5 would make the catalytic domain. And there's only one catalytic catalytic domain? It's the domain that adds the glucuronate to the substrate. But we have different substrates. Now, exon 1 can be different as well. It can produce different substrate binding domains, different domains that can bind to different drugs. How is that possible? Well, it turns out that actually exon 1 is not really a single exon. It's actually made of 10 different exons. Okay? 10 different exons. And, um, sorry, 9 different exons. All right? Now, each one of these each one of these exons has its own promoter. So it can be transcribed independently of all of the other sub-exon ones. Okay? And we can generate 9 different enzymes with 9 different substrate binding sites, but one catalytic domain. So here's how it looks like. So these are the exons that I talked about, 2 to 5, 2, 3, 4, and 5. And these in blue are the exon 1s, and each one of them has its own promoter region. So we can have 9 different transcripts, and each one of them has its own substrate binding domain, but they all have the same catalytic domain. So they can bind to different substrates because of these different exons. That is why in different tissues there are different genes or different proteins that can be produced from this one single gene, and that increases diversity again, and that's what makes us humans. So you have this gene, for example, that can be found in biliary tissue, colon, intestine, liver, stomach, and this enzyme that is produced from this gene right here would target etopocytes. Okay, tamoxifen is targeted by UGT1A4, that is exon 4 of exon 1. Okay, so and it is produced in biliary tissue, colon, intestine, and liver as well. Again, do not memorize this table. It's just to show you again diversity. The last example is a combination of both alternative splicing and alternative polyadenylation. Okay, how does it work? Well, we have a gene that looks like this. Now, if you look at the gene, we're talking about this exon right here. That's an exon. And here we have another exon. And this right here is a region that is part of the exon, but it's not translated. We'll talk about that later. Okay. Here we have an exon right here, and here we have this is part of the exon, but it's not translated. Same thing here. That's an exon, and this whole thing is an exon, but it's not translated. Okay, we'll talk about that in the translation lecture. But the idea here is that we can have alternative splicing. So, for example, we can connect this exon. Oh, and these right here are the introns right here. So that's an intron, that's an intron, this is an intron, and so on. Okay, so we can have alternative splicing. We can connect this exon with this exon right here, okay, which ends there. or we can connect this exon with this exon right here. Okay, this whole thing, by the way. Okay, so we can have, note that we can have we can have alternative splicing producing different messenger RNA molecules. Or we can have this exon connected to this exon connected to this exon, and this whole thing is part of the red exon. Now, except that we can have transcription terminated right here. So we have different poly A sites within the same exon. We can have a poly A signal right here, or we can have a distal poly A signal right here. So transcription can be terminated here, or it can be terminated there within the same exon. Now for this exon right here, termination of transcription is right here. If we're talking about splicing this exon with that exon, termination of transcription would end right here, is located right here. Okay, so we can have alternative splicing of that gene, or we can have different poly A sites. Okay, why is it that we have a longer exon or a shorter exon? Well, we'll talk about that later on when we talk about microRNA and the importance of microRNA in regulating messenger RNA molecules and in regulating translation. So keep that in mind, please. So right here, we can have an example of a gene that can undergo alternative splicing, or it can have different transcription termination sites, polyadenylation signal sequences. Okay, and that is important for regulating messenger RNA and translation. Allah